All right, I think we're going to get started. I have a couple of housekeeping things before I do an introduction. So the CME code for today's presentation, 7185, and that'll be repeated at the end. Also, for questions from Zoom, if you can please put them in the Q&A function, the Q&A section, and not in the chat, please, and then I'll be moderating that at the end. And so I'm very pleased to introduce today's speaker, Dr. Guillermo Horga from Columbia University. He is a dual threat with an MD from Miguel Hernandez University and clinical training as a psychiatrist, as well as a PhD in neuroscience from the University of Barcelona. He completed his postdoctoral research fellowship at Columbia, where he focused on advanced MRI and cognitive neuroscience methods under the mentorship of Bradley Peterson and Anissa Abidargam. And he's currently an Associate Professor of Psychiatry at Columbia and the New York State Psychiatric Institute, as well as co-director of the Clinical Cognitive Computational Neuroscience Center, which I practice saying, at Columbia, and a member of the Adult Lifespan Psychopathology Study Section at the NIH. His research program is focused on neurobiology, neurobiological and computational mechanisms of psychotic symptoms in schizophrenia and related cognitive functions in health, including inference learning and decision-making. And to this end, his laboratory uses behavioral paradigms and computational tools, along with a variety of functional, structural and molecular neuroimaging techniques, especially functional MRI, in healthy folks and in people with psychotic disorders. This work has been continuously funded through private foundations and the NIH. And today he will tell us about his work on neurocomputational mechanisms and neuroimaging biomarkers of psychosis. Please join me in welcoming Dr. Horga.
Thank you so much, Michael Paul, for the kind of introduction and for the invitation to present here today. It's my pleasure to tell you about the work that we've been doing in these areas of neurocomputational mechanisms and biomarkers of psychosis. Can you hear me well? All right, so there's some disclosures that are relevant to the second part of the talk. And like I was saying to some colleagues I met with, I like dividing the work that we do in the lab into two broad areas. One is mechanistic research, the other one is biomarker discovery. And we often merge the two, think of them interchangeably, but I think they're distinct, even though they have clear interrelationships and overlap. But the goals are distinct, and the mechanistic research that we do in my lab is focused on trying to understand mechanisms leading to the generation and expression of positive symptoms. Today I'll mostly talk about hallucinations. The second, biomarker discovery, is related, but ultimately we think of it as trying to develop tools that will be of clinical utility. So it's a little less about mechanisms, more about practical use. And for that, we've been doing work related to this modality of neuromelanin-sensitive MRI that I'll talk about in the last third of the talk. Please interrupt me if you have questions, however you want to do it. It seems like we have enough time. The premise for a lot of the work that I'll discuss today is the well-known relationship between elevations in striatal dopamine and the expression of positive symptoms of psychosis, such as hallucinations and delusions. So I won't be talking about schizophrenia as a whole. I'll focus on the symptom domain of positive symptoms, and in particular hallucinations, which are defined as false percepts without corresponding stimuli. You probably know that in schizophrenia it's very common that people hear voices in the absence of speech stimuli. And so we know that people who have more severe symptoms in this domain, when they have an exacerbation of those symptoms, we see higher dopamine levels in parts of the striatum. We know that currently efficacious antipsychotics act by blocking dopamine receptors. So this relationship is well established, but what's less clear is what are cognitive computational mechanisms linking this to or bridging this neurobiological alteration and the experience of these psychotic symptoms. So I'll be discussing one candidate cognitive mechanism, which is alterations in perceptual inference as a candidate mechanism bridging this neurobiology experience gap. And in particular, I'll focus on hallucinations. So first, what do we know about dopamine in the brain and the type of circuits that seem to be influenced by dopamine? We know that there are parallel loops in the brain, different circuits observe different functions. Early models of psychosis focus more on the limbic system and dopamine in the ventral striatum as part of that limbic circuit. But more and more, there's evidence suggesting a predominant involvement of dopamine in the associative circuit in psychosis. And this is dopamine in particular in the associative dorsal striatum as opposed to ventral striatum. So dopamine in this area seems to be higher in people with more severe psychosis or with exacerbations of psychosis. And this is a part of the striatum which receives specific projections from part of the substantia nigra pars compacta, ventral parts of the compacta, and also receives cortical projections from association cortices, including prefrontal cortex such as DLPFC, dorsal anterior cingulate, but also association regions of sensory cortex, such as association auditory cortex, including the speech selective regions in the human brain, and posterior parietal cortex. So we have known this for some time, and we wanted to think about a cognitive function that is relevant to this circuit and to dopamine in this circuit. And we started focusing on perceptual inference, which is one of the functions observed by the circuit, partly because it's relevant to the perceptual nature of the symptoms that we're investigating, like hallucinations, and partly based on a model that I'll describe. So what is perceptual inference? The basic notion here is that perception is not just a passive process of detecting sensory stimuli from the external environment, because these sensory inputs from the environment are always conveyed with some level of uncertainty or noise. And our brains have developed ways to resolve some of that uncertainty by leveraging our prior knowledge. So doing that generates estimates about the external states of the world, and we call those estimates inferences. Another way to think about this is we can, from a Bayesian inference perspective, we can think about perception as the generation of a posterior belief. This is a probabilistic quantity. And this is simply the combination or a weighted sum between sensory data, the objective sensory data from the external environment, and our prior knowledge. And by prior knowledge, I mean implicit prior expectations that are derived from our previous experience. And the combination of these two, rather than the sensory data alone, is what probably determines our perceptual experience. We can also think of this as an optimal combination of two sources of information with different noise around them. So one is this sensory input in blue, as a Gaussian distribution here with some uncertainty around it. And the other one is the prior expectations with some uncertainty. And we can think about perceptual inference as the combination of those two, depending on their relative level of uncertainty. So giving more weight to the more certain source of information is what will determine whether one or the other plays a bigger role in our perception. So from this perspective, it might be easy to think about ways to bias this process. And Carl Friston proposed a model where a particular bias in perceptual inference could determine hallucinatory percepts. So he suggested that underestimation of uncertainty in prior expectations would lead to excessive weighting of prior expectations on perception and to percepts that would be biased more towards prior expectations and biased away from the objective sensory data. And this could be a hallucinatory percept. So this is a type of prior biased inference where the prior is given excessive weight relative to the objective sensory information. So we found this theory very appealing, this algorithmic model, and we set out to test it empirically. And this is work with my former postdoc, Cliff Cassidy. And we came up with a task to do that, which is a tone duration reproduction task, where people are asked to reproduce the duration of pure target tones. So they listen to a tone and they're supposed to reproduce its duration by stopping a playback of the tone when it matches the perceived duration as they heard it. Critically, those target tones were preceded by context tones that could be longer, shorter, the same duration as the target tone. And we also manipulated the variability in the context tones. The idea here was that we wanted to induce an illusion of tone duration, such that the perceived tone duration would be dependent on the context mean, and that we would manipulate the strength of that illusion by changing the variability in the context tones. And I'll give you an example of this from one subject. So this is data from one subject. All of these tones are 700 milliseconds, so they're the exact same duration. And we can see the reproduction duration on the y-axis. So we can see that this subject is reproducing those tones as longer when they're preceded by longer context tones and shorter when they're preceded by shorter context tones. So this is the illusion of tone duration that I was mentioning. The context is manipulating how the tone is perceived. And this is consistent with this model of perceptual inference, because in this context, which is a low variance context, there should be a fairly certain expectation of the tone based on the context. And when it's combined with the actual sensory input, which is 700 milliseconds, it should lead to a percept that is somewhat intermediate between the two, between the prior expectations derived by the context and the actual presented duration. According to this model, also, this should change when there's more context variability. So in a situation with higher context variance, there should be an estimate of the uncertainty about those prior expectations that should render those expectations less influential on perception. So in the high variance context, we should see a flattening of those slopes that reflect the illusion strength. So the difference between these two slopes. The steeper positive slope is a stronger illusion, and then the flatter one is a weaker illusion, which is what we expect in this context from perceptual inference models. And it is precisely this second part, which according to Friston's model, should be disrupted in hallucinations. If people with more severe hallucinations have systematic underestimation of the uncertainty of priors, they should treat these two contexts as more similar. And even in the high variance context, they should have strong prior expectations with inappropriately high certainty that should still drive perception. So we would expect a smaller change, a lesser uncertainty adjustment between these two conditions. So we had this prediction from the model. We wanted to test this in patients. So we ran this task in patients with schizophrenia who were medication-free and who had different levels of hallucination severity. And what we saw is that those with more severe hallucinations in the y-axis here indeed had lesser uncertainty adjustment. And this was a clinically specific effect in that we didn't see this for other symptom domains, for negative symptoms, cognitive impairment. We didn't even see it for delusions. We saw it specifically for hallucinations and even controlling for those other domains. And it was also cognitively specific in that people with more severe hallucinations didn't have broad impairments of behavior in this task. They didn't reproduce tone duration worse in general. We had some catch trials to measure that. But it was specifically the adjustment in the illusion strength as a function of context variability, which was disrupted. So they showed stronger illusions and illusions that remain strong under more context variance. That's what this is showing. Consistent with the idea that they have this type of prior bias perceptual inference, perhaps related to this underestimation of uncertainty in the priors. The question now is this a dopaminergic phenotype? Is this related to the known elevation of dopamine in dorsal associative striatum? So to answer that question, we performed a PET experiment measuring dopamine release in these parts of dorsal associative striatum, which are linked consistently to severity of positive symptoms. In this sample, we saw also a correlation between dopamine release in this area and hallucination severity specifically. So we measure binding potential with a D2 tracer radical pride at baseline and then post-amphetamine, and we calculate the difference in binding potential, which gives us a measure of amphetamine-induced release, and we call that dopamine release capacity. That's on the x-axis here. We wanted to see if more dopamine release in this area of the striatum would be related to lesser uncertainty adjustment, which is what we saw in relation to hallucinations. And this is what we saw. And we saw this actually in patients and in controls. You might be concerned about some circularity issues because we know that these two measures are related to hallucinations. So maybe they would be related to each other. But we actually saw this relationship also in controls who didn't have hallucinations, suggesting that in general, dopamine in this dorsal associative part of the striatum is related to perceptual inference and is adjustments based on contextual variability that affect the uncertainty in prior expectations. So this is overall consistent with this model that perhaps dopamine elevations in these dorsal parts of associative striatum could be driving this prior bias in perceptual inference, which in turn leads to hallucinations. So we have a more specific form of perceptual bias that we think is relevant to hallucinations. We have also proposed underlying neurobiological circuit that might be implementing this type of bias in the brain. In particular, I have been talking about the dorsal associative parts of the striatum here and dopamine elevation in those parts of the striatum. So I only showed you correlational data. We cannot show that dopamine elevations there would cause this type of prior bias in perceptual inference, but with this more concrete anatomical model and this idea of the type of bias that we expect, we can do preclinical studies. And this is what Katarina Schmack and Adam Kepex did in this paper. They actually performed an optogenetic manipulation that elevated dopamine specifically in dorsal parts of the striatum and dorsal sensory parts of the striatum, the tail of the striatum in this case. And they showed that during an auditory signal detection task, that dopamine elevation would drive a bias in perception that could be interpreted as a prior bias in perceptual inference. They actually measured an increase in false alarms with high confidence, which they considered a hallucination-like percept. So this is consistent with the model, in particular with the idea that dopamine elevation in this part of the striatum could actually be driving the perceptual bias. As you can see here in the schematic, in the circuit schematic, it prominently features parts of the temporal cortex. In the association temporal cortex, these areas of superior temporal gyrus and superior temporal sulcus, which in humans include speech-sensitive parts of the temporal cortex. And we included that here in the schematic because it is known that in people with hallucinations, there's engagement of those regions. There's increased baseline activity when they're hearing voices and at baseline. So there are symptom capture studies with fMRI where people are asked, patients with schizophrenia are asked to report when they're hearing voices, the onset and offset of the hallucinatory percept. And if you look at fMRI signal that changes during that process, during that period, it's generally elevated in auditory cortex and in association auditory cortex, that areas that are speech sensitive regions of the temporal cortex. I'm also showing you an example here of a different type of study that was simply an FDG PET study in first episode schizophrenia in antipsychotic naive patients, where we simply compared the baseline glucose metabolism in people with prominent hallucinations compared to those without hallucinations. And what we saw is that hallucinators had this elevation in glucose metabolism along this association auditory cortex regions, again, in these speech-sensitive regions of auditory cortex. We've also done some other fMRI studies focused on the modulation of activity in these regions as a function of prior expectations. And we focus specifically on this quantity called the sensory prediction error, which is simply the difference between a stimulus and the expectation for that stimulus, or the prior expectation for that stimulus. In this context, the expectation based on stimulus history, whether you've been exposed a lot to that type of stimulus or not in previous trials. And we've looked at this in the context of a very simple task, which is a speech discrimination task, where we simply ask people to report, did you just hear a voice or not? And we present different types of stimuli. We present speech, different sentences, non-speech, other type of auditory stimuli, or no stimulus, a blank trial. And what we have seen in the context of this paradigm, and critically, we manipulate the blockwise probability of those different stimuli in different parts of the task to manipulate the expectation that would be derived from the proportion of trials in a particular part of the task. And what we have seen is that activation in association auditory cortex can be characterized as a type of sensory prediction error. I'll show you more data on this later. But basically, it's a type of sensory surprise, and this signal seems to be weaker in patients with schizophrenia. We compare patients with schizophrenia with severe hallucinations to controls, we see weaker sensory prediction errors. How is this related to the previous framework that I was mentioning before? I was framing perception as this posterior belief, which combines the sensory input and prior expectations. We can simply rearrange this formula to show that we can think about perception as a type of change in beliefs from the prior to the posterior. And that is the same as saying there's a type of weighted sensory prediction error that reflects this change in belief from the prior to the posterior. And according to the model that I was discussing, this model with a stronger prior effect or underestimation of uncertainty around the prior, you can see that the change between the red and the purple distributions is smaller, suggesting that under this model we expect a smaller belief update and a smaller weighted prediction error. So we think that the results that I just showed you of weaker prediction error in patients might be consistent with this type of prior bias in perceptual inference. So I haven't presented a lot of data, but I give you a flavor of why we think that the associative auditory cortex might be involved. There's increased activity. There's this change in the effect of expectations in this region, in schizophrenia at least. So one question is this the only possible model? Is this disruption in perceptual inference necessarily related to this corticostriatal circuit and dopamine in these parts of the association corticostriatal circuit? And the answer is no. We know from more basic preclinical studies that indeed we see this type of sensory prediction error signals in the cortex, in auditory cortex, visual cortex, in different species. I'm showing you here an example in marmosets. We're making different calls, and the recordings are showing activity in auditory cortex. And these auditory cortex neurons are suppressed when the marmoset makes a call, but when the feedback related to the call is distorted, the activity is increased, suggesting that some of that activity is encoding this type of sensory surprise consistent with the sensory prediction error. And there are a lot more recent studies showing that both in visual cortex and in auditory cortex in mice during different paradigms, we also see some neurons that are consistent with sensory prediction error neurons. The laminar profile of those types of neurons tends to show that specifically in superficial layers of cortex, we see this type of sensory prediction error response. And this is consistent with a lot of theoretical work on predictive coding, suggesting that the computation of this type of sensory prediction errors might actually be through a cortical circuit. So you have superficial pyramidal glutamatergic neurons, which receive inputs maybe from the thalamus, and then receive feedback projections that through an inhibitory mechanism might suppress part of that input signal. And that gives you the sensory prediction error. But the important thing is that a lot of the work in this area thinks about the computation of these sensory prediction errors as a cortical mechanism. So it doesn't talk about striatum at all, doesn't talk about dopamine. So this is at least an alternative mechanism that it might be just an intracortical cortical circuit process that might depend on the glutamate and excitation inhibition balance in the cortex, which, as you know, is another predominant theory of schizophrenia, in particular the NMDA hypofunction theory of schizophrenia. So in the study that I'll talk about now, we wanted to consider different upstream mechanisms that might be combined to disrupt this type of perceptual inference signals. So we measured striatal dopamine signals, and we also focused on cortical glutamatergic alterations consistent with this NMDA hypofunction. We also wanted to measure this type of bias in perceptual inference through this type of sensory prediction error signals, because we think that this gives us an index of probably the same process or a related process, but we can measure that directly through neural activation rather than inferring it through behavior, which has some advantages. But we wanted, in this case, to try to link that phenotype of weaker prediction errors to hallucinations, which in the previous work we couldn't do because it was a smaller sample. It didn't have enough variability in hallucination severity. All right, so this is a study that we're currently writing up, which is a collaboration with Stony Brook, with Anissa Abidargam in particular and other colleagues there, and for which my student, Justin Bach, took the lead on, and he'll be defending soon and looking for a postdoc position. He's extremely talented, so I would highly recommend him if you're looking for a postdoc. All right. So sorry if this part is rough. It's the first time that I present this, and maybe the figures are not super clear, but I'll try my best. So this is the task that we use here. The fMRI task is a variant of the task that I told you about before. It's a speech discrimination task. People have to tell us if they heard voices at the end of each trial. And we present speech, non-speech, or silent stimuli. We also use a cluster temporal acquisition design where stimuli are presented in the absence of scanner gradient noise, so that they're very clear. They're all super threshold, so there's no perceptual ambiguity in this task. It's not the stimuli don't have background noise, basically. And despite that, people with hallucinations sometimes make reports of hearing voices in the complete absence of any stimulus, and we call those auditory verbal hallucination or AVH trials. Like I said before, we manipulate the probability of these different stimuli over different blocks of the task. So the speech, non-speech, and silent trials have different proportions in different parts of the task to manipulate the expectation for that stimulus type over the trials. And we collected data from this task in a population of controls. We're calling them non-psychotic individuals here, and also people with schizophrenia spectrum disorders, which we're calling psychotic individuals here. And critically, they were unmedicated, and they had a range of severity of hallucinations from very low to very high severity. The first thing that we showed here, like we showed in our previous work, is that the rate of task AVH, or these reports of hearing voices during silent trials, is correlated with hallucination severity measured clinically. So it seems like this task is tapping into something that is relevant to their hallucinations outside of the research paradigm. And not only that, but the hallucination rate or the AVH rate tends to be higher in blocks with higher probability of speech. So it seems like they're integrating some of this context and the expectations generated by the proportion of speech trials in the task, and that is influencing if they report hearing voices in different parts of the task. So the first thing that we wanted to do was to characterize better this response, the stimulus evoked responses in the association auditory cortex. And we focused on these speech selective regions of auditory cortex, which we can isolate by this contrast of speech versus non-speech. And these are the well-known temporal voice areas in the human brain. By definition, we expect to see a stronger response to speech than non-speech, and these areas tend to respond to auditory stimuli in general, so that's not surprising. But we wanted to see, will we see decreased activation when the expectation for stimuli is higher? So when the blockwise probability for that stimulus is higher, do we tend to see a suppression of activity? And that's what I'm showing you here in this model agnostic analysis. And we see that in blocks with higher probability of stimulus, for instance, even during the presentation of the stimulus, the response is weaker. So suggesting this modulation by expectation consistent with the sensory prediction error. We also actually fitted a computational model to the trial by trial activation in that region. With this task is strong enough to actually fit a model, unlike some other cognitive reward tasks. But it's a very simple model that it's a combination of two different types of sensory prediction errors, a stimulus general prediction error, this beta stimulus, and a speech selective sensory prediction error, the beta speech, and it also has a shared learning rate. We did a lot of model comparison, and this was the winning model, and it seemed to explain a lot of the variance in the auditory cortex signal. I'm showing you here basically the relationship between model activity and the actual activity or the fMRI signal in these regions. And you can see that the model does a pretty good job at capturing variance over different trials. And this is the average across subjects for a given trial type. But you can see that it obviously separates activity from speech and non-speech to silent trials, which that's easy. But within a particular trial category, it explains some of the variance, and that variance has to do with the expectation of that type of trial and that part of the task. I told you earlier that a sensory prediction error is basically the difference between stimulus and the stimulus expectation. And that makes a particular prediction of the activity profile that we should see in a region that is encoding a sensory prediction error. And that is a combination of two main effects, if you want to think about it from a statistical perspective. First, activity should be higher when there's a stimulus presented than when there's no stimulus. And we see this in this plot here. In general, activity is higher for speech and non-speech compared to the silent trials. And there should also be a negative effect of the expectation of the stimulus, which is the x-axis here, the model probability of the stimulus. And that's the negative slope that we see in all of the lines. So that is the profile that we expect for a sensory prediction, or the combination of the separation of the lines and the negative slope. And this is what we're seeing in the raw data. You can see the shading is the model, but it fits pretty well with the data. So we think activity in this region is well characterized as a combination of sensory prediction errors for different features, in general for the stimulus, auditory stimulus, and then in addition, there's a sensory prediction error for speech. I won't go into all of the details. I was talking to Caroline earlier about this. We think that it's very important to characterize these signals and to make sure that we can recover them well, that they have good enough reliability, that we can interpret into individual difference. So we did a lot of analysis to convince ourselves that we could do that, and I can discuss that. But after having done that, we wanted to see essentially, are these type of sensory prediction errors different as a function of severity of hallucinations? That was the main question here. And of the two types of sensory prediction errors, we saw that the stimulus general sensory prediction error, the strength of that type of sensory prediction error, was indeed correlated with the severity of hallucinations. So people with more severe hallucinations had weaker sensory prediction errors in the association auditory cortex. We also saw that effect for the task AVH rate. So for hallucination severity measured clinically through the PANSS, or the task AVH rate. And that effect, like before, was clinically specific in that we didn't see that for other symptom domains. We actually saw it for delusions, but that went away after controlling for hallucinations. And the effect of hallucinations didn't go away with control for delusions and all of these different severity of these different symptom domains. So we think that this is a fairly specific phenotype that relates to hallucinations. So people with more severe hallucinations show this type of neural alteration in inference signals, this weakening in sensory prediction errors, which seems to be selectively associated. Now the question is, what are potential upstream mechanisms that might lead to this disruption in this type of inference signals? So we wanted to consider different candidate models. One of them in green here is the one that I was talking about before, this corticostriatal loop and dopamine dysregulation in the dorsal associative parts of the striatum. So we here measured again dopamine release capacity. In this case, we used a different tracer, PHNO, but we did the amphetamine challenge as before, and we measured dopamine release capacity. We also wanted to consider this other hypothesis, which I would probably better call the cortico-cortical circuit. And we measured glutamate or an index of glutamate function in the same region of auditory cortex with MR spectroscopy. And we measured GLX, which is a combination of glutamate and glutamine in that region. And then we considered a third hypothesis because there's some preclinical models suggesting that maybe some of the auditory dysfunction in schizophrenia might have to do with altered thalamocortical input. So for that, we measured resting state connectivity between the auditory thalamus and the early auditory cortex using first a validated thalamic localizer task that isolates the auditory thalamus. So we use this localizer to find the auditory thalamus and then measure the connectivity between there and the early auditory cortex. So we considered these three potential upstream mechanisms for candidates that would lead to the disruption of these inferential signals, these weaker sensory prediction error signals in the auditory cortex. First, we saw that of these three measures related to these three candidate mechanisms, two increased dopamine release capacity in the associative striatum and increased thalamocortical connectivity were related to the symptoms themselves, were related to hallucinations. We didn't see an effect of auditory cortex GLX, but there's a previous paper that has shown that effect. We don't see it here. But the key question was, again, of these three measures, which ones are related to weaker sensory prediction errors? So we collected in a multimodal cohort, including people with schizophrenia and healthy individuals. We collected these three measures and the task fMRI measure of sensory prediction errors, and looked at the relationship between sensory prediction error strength and these measures. And what we saw is that higher dopamine release capacity in associative striatum was related to weaker sensory prediction errors, but the other two measures were not related, either in independent models, just with one measure at a time, or in this combined model with the three measures. And what we're predicting here is the strength of the stimulus general sensory prediction error, which we're calling beta stimulus here. We also looked at everyone who had PET and task-based fMRI data, which we're calling PET cohort here, and saw that in that cohort, which is a little larger, higher dopamine release capacity again was related to weaker sensory prediction errors, but only in the associative dorsal striatum. In a model that included associative dorsal striatum and ventral striatum and extrastriatal regions, only the associative striatum dopamine was related to weaker sensory prediction errors. We also wanted to look at the interactions between the relevant striatal regions and the associative auditory cortex regions in terms of their functional coupling, because in this model where we have measured sensory prediction errors in the auditory cortex, dopamine release in the striatum, but we're not really speaking to how they might be interacting with each other. We wanted to look at the functional interaction between those, and we wanted to look at this over trials because we're particularly interested in expectation modulations, which reflect the incorporation of information over different trials. So we did this type of cross-lag functional coupling analysis. The first thing that we saw is that if we look at auditory cortex activity in the current moment and see how that depends on previous auditory cortex activity and previous striatal activity, we see an effect of both. So auditory cortex depends on both past striatal and past auditory cortex activity. And we see this separately in healthy individuals and in the schizophrenia cohort. We wanted to see how these different paths would be modulated by dopamine release and by hallucination severity. And what we found is that this path between past striatal activity and current auditory cortex activity was weaker in people with more severe hallucinations and was also weaker in people with more dopamine release capacity. And these are combined, but these are partly different cohorts. Overall, consistent with the idea that striatal dopamine excess in these dorsal associative parts of the striatum and altered corticostriatal interactions in the associative circuit could be driving this altered neural inference signals, these weaker sensory prediction errors in auditory cortex, also as part of this association circuit, leading in turn to hallucinations. So at least consistent with this model. So we're not ruling out, of course, other contributions of cortical glutamatergic dysfunction. We just had this one measure, and we actually had some evidence that would be consistent with an additional contribution, which I can come back to if you are curious. But in general, we find evidence that there's an involvement of this corticostriatal circuit and dopamine elevation in the striatal parts of the circuit in disrupting this type of perceptual inference signals and producing a prior bias form of perceptual inference that seems to be relevant specifically to hallucinations. So this is an interim summary of this part. I've shown you that prior bias in perceptual inference seems to be selectively related to hallucinations, that what we think of as a proxy for that in terms of decreased sensory prediction errors in associational auditory cortex seems to also be selectively related to hallucination severity, and that this form of biased perceptual inference seems to depend on dopamine elevations in dorsal associative striatum. And we also found some evidence for dysfunctional interactions in association corticostriatal circuit as part of this mechanism of hallucinations. How am I doing on time? Okay. All right, so questions on this part? I threw a lot at you, but
you said, and maybe you want to save this for the end, but that you did a lot of work to kind of convince yourself about the reliability and the validity of the model, and that that it was robust. And I wonder if you want to say anything more about that, especially for folks who are kind of new to modeling and kind of interested in learning. That question always comes up. How do I know I can trust this? So. If there's anything, you can talk about it more at the end if you prefer.
Maybe at the end might be better, because sure. I could talk about that for a while.
Dig that. Yeah, sure. Right. Okay. Thanks.
But yeah, come back to that. Other clarification questions or anything?
So it's like more correction error also and yeah. You can repeat the question.
I think the question is, how do you reconcile this with findings of a blunted reward prediction error in schizophrenia? Does that?
Yes. So my understanding is that increased dopamine can be related to increased prediction error signal, especially in the reward context. And you mentioned that, but actually the increased dopamine is related to increased prediction error signal in schizophrenia, especially in striatum. So yeah, my question is how to reconcile the increased dopamine and increased prediction error.
Yeah, that's a great question. So what we had hypothesized is that maybe there would be a specific, more than just elevations in sensory prediction errors, that there would be a bias towards positive prediction errors. We're not as confident that we can measure that in this signal, because that requires a more complex model. But there are two aspects to that. The known reduction in reward prediction errors in schizophrenia, that's more of a schizophrenia general phenotype rather than a positive symptom phenotype. Here we think that this decrease in sensory prediction errors I take to be a disruption in this system, and we're measuring this only at the level of the sensory, the association auditory cortex. So what's happening at the level of dopamine, what's happening in the striatum, and how does that disrupt this type of signals in the cortex? There are a lot of kind of steps there, and we don't have a super clear idea yet. But this wouldn't be directly a dopaminergic sensory prediction error signal. So maybe the disruption in dopamine in the striatum would produce a downstream effect in this type of computation in the cortex. So how we're thinking about it. And we see some evidence for some sort of competitive interactions between what's happening in the striatum and in the auditory cortex. So there could be different systems computing in different types of sensory prediction errors in parallel that kind of talk to each other, and it seems like that interaction might be disrupted. All right, so I promised that I would talk about neuromelanin-sensitive MRI, and here in this last part of the talk, I wanted to shift gears and talk about biomarker discovery, again with the goal of ultimately developing tools that could inform clinical practice. So for this work, we've focused on this modality of neuromelanin-sensitive MRI. So I thought I would give some background first about neuromelanin, which is a pigmented product of dopamine metabolism, which is generated through oxidation, which we know accumulates in the form of neuromelanin iron complexes in the body of dopaminergic cells in the substantia nigra, pars compacta mainly. We also know from basic studies, including from our collaborator, Dave Sulzer, that increasing pharmacologically dopamine synthesis leads to more dopamine intracellularly in the cytosol, and that leads to more synthesis of neuromelanin. We also know that neuromelanin iron complexes are paramagnetic, and they can be imaged with this family of sequences that we call neuromelanin-sensitive MRI, which is a combination of T1 and magnetization transfer contrasts. So putting this all together and some of what I mentioned before, our hypothesis here was that in people with psychosis who presumably have elevated dopamine synthesis in these dopamine neurons, we should see more accumulation of neuromelanin, and we might be able to pick that up with neuromelanin-sensitive MRI as an increase in the contrast. So with that hypothesis in mind, we started collecting data with neuromelanin MRI data, which looks like this. You can see here a mask of the substantia nigra in yellow that actually includes some parts of the VTA in the medial portions of that mask. So we look at the signal in those voxels normalized by the signal in the cross-cerebri magenta reference region here, which is a region that has negligible concentration of neuromelanin. And we look at that contrast in a voxel-wise manner, partly because we have very high spatial in-plane resolution here with this technique. It's less than 0.5 millimeters. So that allows us to look at spatial patterns even within this nucleus, which is a pretty small nucleus in the midbrain. And we only image a slab here in the brainstem. So the first question was, is the contrast in this region, the substantia nigra, VTA, or parts of the substantia nigra related to dopamine function? So we measured dopamine release capacity with the methods that I discussed before in dorsal associated striatum again, and saw that those people with higher dopamine release also have higher neuromelanin MRI contrast, suggesting that the neuromelanin contrast could be a decent proxy for dopamine function across subjects. We then had this hypothesis that people with more severe psychosis, who tend to have more dopamine, would also have elevations in neuromelanin MRI contrast, at least in some parts of the nigra. And we saw in a cohort of people with schizophrenia who were medication-free and also med-free individuals at clinical high risk, that those with more severe psychotic symptoms, more severe positive symptoms, or more severe attenuated positive symptoms in the CHR tended to have higher neuromelanin MRI contrast in these parts of the nigra that are highlighted here. And that's interesting in particular because we found this predominant effect in the ventral parts of the nigra pars compacta, which are the parts of the dopamine where the dopamine neurons live that project to the dorsal associated striatum. So this is consistent with the specific circuitry that I was mentioning before. But we were very excited to see these effects, and we've done, since this paper, we've done a significant amount of work in this area. And the summary for a lot of this work is that we think that there are desirable properties in neuromelanin MRI that make for a good candidate biomarker for psychosis. And I'm summarizing them here. One is that it's very easy to acquire. It's a non-invasive technique, so it's much easier, for instance, to do than a PET scan, especially 2 PET scans if you do the amphetamine challenge. We have some data supporting internal validity as a measure of neuromelanin and as a measure of dopamine function. And we've also evaluated the reliability, which is excellent, and that was already known, but we replicated even for voxel-wise analysis. And we also have some data supporting the scalability of these measures. So for instance, we've developed standardized acquisition protocols, and we have some data supporting the ability to harmonize data across different scanners. So all of these, again, desirable properties if you want to develop a biomarker. The question is, what could this be used for in the clinic? So we think the more promising applications would be as a predictive biomarker. I think that clinicians mostly want to know things like, okay, can you predict, can you prognosticate the outcome in someone at clinical high risk? Can you tell if that person will develop a psychotic disorder? Or can you tell if this person in their first episode of psychosis will respond well to first-line antipsychotic treatment? And if you're very confident that they wouldn't, maybe you want to consider alternative medications, maybe xanomeline, maybe something else. So those are the type of questions that which we think are more relevant perhaps in the future in clinical practice. So we wanted to start getting into some of these questions, and to make predictions, the first thing you need is ability to generalize results to new individuals. Any biomarker needs to make accurate predictions, or at least above chance predictions, in new individuals, in a new cohort, and the new patient that you have in front of you. So we wanted to test the ability to make predictions. The first thing that I'm not showing you here is that we replicated the elevation of neuromelanin MRI contrast in those with more severe positive symptoms in schizophrenia in a new cohort here. But the main question was about prediction. So what we did to test prediction is we used this machine learning approach that uses support vector regression, leveraging the contrast across different voxels to try to predict severity of psychosis in out-of-sample individuals. So we trained an SBR model in 9/10 of the data and then tested the performance in the left-out 10th and then iterated. And this is what's called cross-validated performance. So when we evaluated that, we saw that predictions were above chance level, both in terms of the error being smaller than expected by chance based on a permutation test, and the strength of the correlation between the predicted level of psychosis and the actual level of psychosis was being also above chance levels based on the permutation test. I told you that in this analysis, we were using voxel-wise patterns, and these are the weights that the model is using to compute essentially a weighted sum of the contrast to make its prediction about the level of psychosis severity. And to really test the generalizability, we want to take this model, freeze these weights, and then apply it to a new cohort without training anymore. And we did this with an external validation cohort that came from our collaborator in Mexico, Camilo de la Fuente. And this was a very different sample in different clinical contexts, antipsychotic naive first episode, different country, different scanner. We just took this model and tried to see what we could predict. And we were very pleasantly surprised to see that we could predict above chance level, barely, but we could. The more important part here is that the effect size was very comparable to that we got in the internal validation sample, suggesting that there's that the model performs accurately, that it doesn't overfit, and that it generalizes at similar levels to the internal validation sample. So this level of prediction performance is not too impressive, but it's a proof of concept that we might be able to develop models that generalize to new cohorts, which is necessary to develop any biomarker. So within there's potential here. However, the question that is of more clinical relevance is not, can I predict the level of psychosis based on neuromelanin MRI scan? It's probably more pragmatic information, such as, is this person going to respond to antipsychotic treatment? So we've started looking at that, not with predictions yet, but just looking at associations in sample. And we try to reproduce what's known with PET studies that show that people who respond better to antipsychotic medication in their first episode tend to have higher dopamine function. So the people with higher dopamine synthesis or release capacity are better responders if you evaluate the clinical response at follow-up. So we use that type of design. We collect neuromelanin MRI data at baseline. People were treated with antipsychotics, and the clinical response was evaluated at 6 months. And this was data collected from Elsmarie van de Giesen in Amsterdam, and we were collaborators in this study. So what we found here was that, as predicted and in line with the PET studies, treatment responders, the first episode patients who responded at follow-up to antipsychotic treatment at baseline had elevations in neuromelanin MRI contrast in the Niagara compared to non-responders and compared to controls. And interestingly, we saw this effect also in the ventral parts of the substantia nigra pars compacta, like the effects of psychosis that I mentioned before. So again, this is not prediction yet. We definitely wouldn't be able to use this in the clinic, but this gives us hope that we might be able to develop predictive models that might generalize to new individuals that could have some relevant implications for clinical practice. And so we want to do that type of study, which would require a much larger sample to train these sort of models. And with that, I'll just summarize what I've said in the first part about mechanistic research. Basically, I told you that prior biased perceptual inference seems to be important for hallucinations, that it depends on dopamine in the associative stratum, and that it seems to involve this association corticostrial circuit, perhaps in addition to other cortical processes and perhaps other mechanisms. So we definitely find an involvement of this associative corticostrial circuit. In the second part, I discussed neuromelanin MRI as a candidate biomarker for psychosis. I showed you some proof of concept evidence for ability to generalize and some initial results on relevance to treatment response. With that, I'd like to thank my lab, collaborators at Columbia, the C3N Center that I co-direct with Gaurav Patel, and collaborators at other institutions, in particular Stony Brook, and funding sources, the NIH especially. And thank you all for your attention. Hopefully we'll have time for questions.
All right, thank you again, Guillermo, for a wonderful talk. I will make sure to put the CME code again in the chat for everyone who needs that. It is 7185 for those of you who are listening. We will take questions both from Zoom and in person. If there's anyone here who has a question, I can certainly begin. I have a number of them. I am wondering about just kind of general methodological considerations for the neuromelanin-sensitive MRI measures. You know, things like the time course, you know, is it particularly slow or fast compared to other functional MRI measures, gradient echo MRI, things like that. Do you need specialized coils, special support from MR physicists in terms of getting things set up just right? Do you need ultra high fields, those kinds of things?
So it's very easy to set it up. I think that it might already be set up in some scanners here, if I'm correct. Anna is nodding. So, and you have like the best physicists here, so you could do anything. But that's easy to do. So yeah, I think that definitely in the scanners you have here, you can do it. Most 3D scanners you can do it. The acquisition, I think you asked about the time scale. The acquisition itself can be around 10 minutes. And were you asking about the time scale of the signal relative to fluctuations in?
Yeah, can you use it functionally, like to observe changes in real time, or is it more like spectroscopy where you're going to sit there for 10 minutes and you get one measure for that entire 10 minutes? Okay, so it's a more kind of static at rest, or maybe throughout the context of an entire task, they're doing it through that whole 10 minutes, but you get a single measurement for a 10-minute period. Got it.
It's a single measure that we interpret as a kind of longer time scale index of dopamine function. There would be even longer time scale than something like a PET measure. With a PET measure, you would also get one. I mean, there's some task-based PET designs, but most typically, like with the amphetamine challenge, you would get one index of how much dopamine release, so like one level of dopamine function. So here would be similar, but keep in mind that this is an indirect measure that probably reflects dopamine function over a certain time, at least probably weeks or months. We don't know exactly, but this is the effect of how dopamine in the cell leads to this conversion to neuromelanin and keeps accumulating progressively, which it accumulates in everyone, but would maybe accumulate a little more in people who have higher dopamine function. So yeah, we think about this as potentially trait-like, or it could be state dependent, but be modulated over relatively long timescales. So I don't think that you could see any changes in a kind of in a short time scale during a paradigm. We haven't tried that. If you saw them, I would assume that it's not actual neuromelanin, but it could be other maybe changes in the cells that affect the signal. If you'd like now or later, I'm happy to take your first question.
Yeah, if you'd like to go back to talking about the model and kind of how you convinced yourself that it was robust in that from the first part of the talk, that'd be great. Yeah.
So I think that especially when you use computational modeling, you want to make sure that you can recover the parameters. So there are analysis called parameter recovery, where you just basically simulate data and try to fit it to make sure that the ground truth, which is the simulated value of the parameter, can be recovered when you fit the data and is not mixed with other parameters. In this case, it's easy because it's continuous data and there are very few parameters, but it's good to check that you don't have parameter trade-off, that you can recover accurately the value of the parameter. Here, we also measure test-retest reliability with ICC. That's good to show that you have good reliability. The other thing that we did that was a little more special, and I think a higher bar, was to show that first that the level of precision in the estimates, the uncertainty around the estimates, was relatively small compared to the range of estimates that we had. Right, we want to have enough variability in the estimates to look at inter-individual differences, and enough precision for a given estimate for a person so that it wouldn't overlap with another estimate. So if the parameter value is 0.5 and the uncertainty around the estimate is 0.01, you're pretty sure that you're around that range and might be very separated from another person that has a much smaller point estimate. But if the uncertainty starts overlapping between different point estimates of the parameter values, then you might not be able to reliably estimate the differences. And the last thing that we did was to show that we can better explain parameters for a single subject than if we use the parameters from other subjects, to really show that you get an advantage in using the subject-specific parameters over parameters from other subjects. And that to us convinces us that these differences are meaningful, that the parameter you get for the subject actually fits the data better for that subject than other parameters. So we did a number of things like that, but yeah, ultimately you just want to have enough range and enough precision in the estimates and enough reliability to interpret differences.
The model just or like use a model to
Yeah, did you feed the model just to behavior data or both to like neural and behavior data?
In this case, we actually did it only to neural data.
Oh, neural data.
We've been playing around with joint fitting of behavior and neural data, but the behavioral data here is not very rich in this paradigm, so you don't get a lot of information. You get a lot more information from the trial by trial changes in the neural data. So it's a type of neurometric fit rather than the standard psychometric fit.
So is that like similar to some Carl Friston's work, like precision predictive coding model for fMRI data?
Yeah, I mean, you can think about these type of models. They're learning models, but they're looking at sensory prediction signals. So yeah, you can think of them as predictive coding models.
Thanks for your talk. Could you explain more about the neuromelanin-sensitive MRI technique? I'm just trying to look at your paper. So is it like a change in contrast that you see? And if so, like, is there anything else that could affect that, like water content or fat content in the tissues?
Yeah, so the normal MRI signal is sensitive to neuromelanin content. We know that, but it's also not specific. So it's, you can think of it like bold fMRI. It's not actually measuring neural activity, but it is sensitive to changes in neural activity. So I think about it that way. Some people have suggested that it's mostly driven by the fact that these dopaminergic neurons are bigger, are pretty large, and have more water content, like you said. We don't think that's the case, that's the only factor, but probably it does contribute, because there's some studies, there's a recent study in rats that did a manipulation, inserted like a human tyrosinase in those neurons in one side and made them produce more neuromelanin in only one side, and you see in that side an increase in the contrast with neuromelanin MRI, but you don't see it in the other side, even though basically the size of the neurons and the water content is controlled. The neuromelanin content itself seems to be driving the signal. But yeah, from this data, we can't say what part of it might be related to water content or what part might be the neuromelanin per se. So it's probably a mixed signal, not perfectly separated. And what we measure is it's basically the signal. It's just that we're normalizing it by the signal in this other region to basically standardize across subjects and have a contrast ratio measure. Does that answer your question?
Yeah, that makes sense. Thank you.
Beautiful work. I'm just really impressed by the fact that you developed this model, this computational model, but then you further tested it to look at individual differences. It's very impressive. The neuromelanin, it seems like you see that as more of a trait biomarker that rather than a state biomarker for a propensity to develop positive symptoms. Is that?
I said that. I would say it's somewhere in between. I think that it captures some aspect of the psychotic state, but maybe not with very high temporal resolution. So we don't, but the jury's out there because the jury's still out on that because we don't have the data. So we don't know really how it changes with treatment, how changes in neuromelanin MRI contrast track exacerbations of the illness. So we haven't done the longitudinal studies to really answer that question. We and others tend to assume that it reflects this long-term index of accumulated dopamine function. So, but because we don't know the exact time scale of that, we don't know if it could track several months of psychosis and then relatively normalize after that if the person becomes asymptomatic. So it's not clear what part of it is more state-like or trait-like, but I think that it should track states to some degree, but probably with a delay, basically over longer time periods. I'm not sure if I'm being clear about that.
Yeah. Okay. And so kind of a related question. So, and one nice thing about the model is that it's relatively specific to a symptom domain, and it's not really confounded by generalized deficits in cognition and so forth, or even other symptoms. So when we think about psychosis that appears in people with bipolar disorder, do you think it's a similar mechanism for them? I mean, for them, it's more state dependent, mood dependent, of course, but do you think it's the same mechanism?
I think so. We don't have data to speak to that, but there are at least some evidence from PET studies suggesting that severity of psychosis in bipolar disorder, just like in schizophrenia, is related to increased dopamine synthesis in similar parts of the striatum. So that the dopaminergic phenotype, I don't think is a diagnosis-specific phenotype. I think that's about positive symptoms. It's not about schizophrenia or bipolar disorder. So at least part of that, and my guess would be part of the cognitive perceptual biases should be similar in bipolar disorder. The only reason why we don't study bipolar disorder is what you mentioned, right? That we think that the psychotic symptoms are going to be more transient and state dependent and seen in an acute manic episode, and it would be particularly hard to study those people right during that episode, we think, or at least we don't have the experience to do that. But we don't hypothesize for any of this to be specific to schizophrenia. Maybe you could test the first part of your question about the trait state distinction in bipolar disorder, depending on how long their episodes are. If they have mood episodes with psychotic symptoms, depending on how long those are, whether you're going to start seeing a normal MRI contrast change. That would be interesting.
Thanks for your talk. I had a question. I was just curious about were you surprised about not finding an association between the sensory prediction error and glutamine signal? And what do you make of that?
So we hypothesized that we would see one, and we didn't see it, so we were surprised in that respect. I think that we don't have enough confidence in the null result because it's not such a big sample to really rule it out. So I think that it's just lack of positive evidence rather than evidence for negative result. One thing that I was going to mention. Sorry. Is that in this analysis, which are sort of preliminary, but when we looked at this functional coupling effects, you can see here that I didn't say it, but this relationship between auditory cortex activity in the past trial and in the current trial, that also seems to be relevant to hallucinations, in addition to the path between the past striatal activity and the current striatal activity and the current auditory cortex activity. So this path here. And when I think about glutamate function, I think about recurrent excitation. So how activity might remain high in auditory or in the cortex as a function of increased excitation relative to inhibition. So this effect, which is in addition to the other effect from past striatal to current auditory cortex, could be an index of that kind of intercortical AI balance effect. That's how we're thinking about it. And we saw some effects that I'm not sure how reliable this is, but some of that effect might actually be related to MRS GLX. So it's not exactly what we were trying to see, this effect directly on the sensory prediction errors, but that might speak to two parallel mechanisms. The story gets complicated because they might actually be, like I was saying earlier, in opposition. So it might be that there's some balance between these two paths, like the weaker the striatal influence, the stronger the auditory to auditory influence. But we haven't finalized that part, so I don't. So there might be some weak evidence for an involvement of both processes there.
Did you look at any other metabolites?
Sorry?
Did you look at any other metabolites like GABA?
We didn't here. We didn't have the appropriate sequence from my understanding. And this was already a super long study because we had 2 PET scans, a task fMRI paradigm, then the thalamic localizer, the resting state scan, whatever. It was a lot of stuff. It was, probably close to how much you can get in a patient with schizophrenia. And these were all medication free, so it was particularly hard.
I wondered if we could go all the way back to the tone estimation task that you talked about at the very beginning. You showed that variance affected the slope in terms of kind of the effect of this illusion across short, medium, and long context. It seemed like there was also an effect there of a change in slope, but also a change in intercept. And I wonder how you think about those two separately or together, and are they both reflecting the strength of this illusion, or is it really mostly the slope? You seem to focus on that.
In this particular task and in this subject, you don't see it in every subject, but in this subject, there's an offset. We think it's a kind of like a non-decision time, like how much they take to actually press the button to say like, this is how long the stimulus was. But when we looked at the intercept, we didn't see effects of psychosis there. In general, they're lower in patients than in controls, but as a function of hallucinations, we don't see a difference in the intercept. So we treated them as dissociable, and it seemed like there was some evidence for that.
So it's more about individual differences and less about the group difference for that kind of non-decision time or whatever that
Yeah.
intercept is. Thanks.
I have a question. Can you talk about or clarify a little bit more about the support vector regression you did, especially voxel-wise? I know that they require lots of iterations of data inputs, and so is it all the voxels that you're using to classify, or do you have some type of trial-wise information that you're also using? I apologize if it's a bit of an ignorant question.
That's a great question. Trying to find the slide, sorry. So what we're doing, we just have for each subject, we just have the contrast ratio map, and we have a measure of severity of symptoms, right? So we don't have trial-wise information. We just have one clinical, one MRI measure. And for the MRI measure, we have actually several values which reflect different voxels. So I think of this as basically you can think of it as a logistic regression where you're predicting, or sorry, in this case it's not logistic, it's just a regular regression, but you have, what you're predicting is the severity of psychosis. And for predictors, you have one predictor per voxel. So you have all of those features that you're using to predict severity of psychosis. But you can't do that with a regular regression. You have too many predictors to fit the model, but you can do that with SVR. But essentially, yeah, it's the combination. The prediction ends up being some weighted sum of the contrast across different voxels with some bias term, and that gives you an index of the severity, the predicted severity of psychosis, and then you just check in the held out data set whether you actually have one prediction per subject, you have one level of severity per subject, and you see if those are correlated, and you measure the error in the prediction. Does that clarify?
I'd like to ask a follow-up question about that. Does that mean, does it suggest to you then that there's some information in the spatial pattern of the neuromelanin signal across this region that is, I saw you kind of showed a map earlier. Is there something there in terms of, specific subregions or how it's distributed across?
Yeah, so it seems like there's some relevant spatial information there. It's a bit hard to interpret. What I can say is that this pattern is more related to this ventral voxels. If you measure the mean signal in the ventral voxels that had the psychosis effect in the previous study, it's related to the predicted psychosis from this pattern. But if you measure the overall mean contrast in the whole mask, it's not related to the prediction from this pattern. So it seems like at least part of the pattern has that kind of topography of being related to the signal, in particular in the ventral voxels, but it seems richer than that. So it seems like there might be a slightly higher dimensional higher dimensional information here spatially that is relevant to the prediction. I don't think it's super high dimensional, so probably if you do PCA on this data, probably there would be a few components that explain the different combinations of patterns, but it seems like it's more than just kind of like the average signal in the region, and that if you actually leverage all of that spatial information, you can predict better. But we haven't really started, you know, for instance, doing ablations of you know, higher level components to see if that the prediction starts getting worse, because we didn't want to, you know, overfit models too much or try too many things. We wanted to do some basic analysis, but I do think there's some spatial information there.
So do you think like I was really impressed by this kind of like dopamine theory of hallucination, and do you expect that this theory can also apply to like delusion as well?
So two things about that. We see that in general, the relationship between dopamine and the symptoms doesn't seem to be specific for hallucinations or delusions. Even in these data here with neuromelanin MRI contrast as a proxy for dopamine, we see that the effect of positive symptoms is driven by both. Doesn't seem to be a difference between delusions and hallucinations. For the other cognitive models and inference models, we've also done work with other tasks that suggest that there's a similar type of prior bias in inference. We see that other people see different things, so there might be less converging than the work in hallucinations, but at least in our work, we found a similar type of prior bias, but in a type of inference that I think of as more abstract or higher level. So instead of making inferences at the level of the stimulus, was the stimulus there or not, it's more like a categorization on top of that. Or an inference on the origin of the stimulus. Is this stimulus coming from this context or this other context, rather than was there a signal or there wasn't a signal? So I think that dopamine might be involved in both of them. It is possible that the disruption happens at one level and sort of propagates to their level, or it's also possible that it affects both levels simultaneously. And I don't think we have an answer for that. But what I can say is that cognitively, the relationships to dopamine, we've found them more reliably for the more basic sensory paradigms and for this lower level of inferences that seems to be more related to hallucinations.
Have you looked at visual hallucinations at all in terms of, you know, you have ratings of, you know, a variety of hallucinations probably. A lot of several folks here do work in visual processes, including myself. And, you know, one thing that we oftentimes see is that people with schizophrenia are less prone to illusions that, you know, are normative. I'm just wondering if you've looked at in that domain at all.
We have, to some degree, in some of the recent studies, including the study with Stony Brook, we collected the chat scale, which measures also, it's kind of like the CIRADS, but includes different domains. So we have a ton of information. It's like very sort of like deeply characterized clinically. And we haven't gone over all of it. So in general, in that cohort, we saw less variability in visual hallucinations. So if we looked at the high severity, low severity, we would have pretty few high severity visual hallucinators. So we might not have the power to really speak to like modality specificity. But yeah, we have a lot of things to do still with those data, looking at kind of specificity to auditory versus visual. We didn't do a visual task, so it might be that there might be a modality specificity on that too. And I think that Al Powers has some data with signal detection paradigms where you condition an auditory signal on a visual cue or the other way around. And it seems like they see this type of exaggerated conditioning effects more for auditory hallucinations on the auditory task and for visual hallucinations in the visual task, but we haven't done that. Part of it, the logistics of just like, stopping somewhere, and because we already have a lot of data loss, and like you can see that the sample sizes are pretty different depending on the sort of which measures we're looking at or how many measures we have in the same subject.
All right, well, shall we thank Dr. Horga again for his very exciting and interesting talk? Thank you very much.